Cells, the living units. You have trillions of these guys in your body, and each one of them behaves like you do. Each one takes in oxygen, puts out carbon dioxide. Each one takes in sugar. Each one has little organelles, that keep them functioning. They are the smallest living unit. So you've probably heard of bacteria. That is a single cell living unit. If you haven't had micro yet, viruses actually aren't alive. They can't live on their own. They must invade a host. But single-celled organisms that you've heard of, like a paramecium or an amoeba or bacteria, E. coli, staph, all those things are single cells, and they live on their own. So that's essentially, in a nutshell, what this chapter is going to go into detail about.
You and your future patients are each made up of trillions of cells, so it's crucial to understand what cells are and how they work. In this chapter, you will learn about the general structure and function of cells. There are over 250 different types of cells in the human body, but all of these cells include three main parts: the plasma membrane, the cytoplasm, and the nucleus. The plasma membrane is the outer boundary of the cell, which contains the cytoplasm, and the nucleus, the cell's control center. The plasma membrane of a healthy cell is a selectively permeable barrier. This means that the plasma membrane allows beneficial substances like nutrients to enter and remain in the cell, while keeping undesirable substances out of the cell. It also keeps valuable proteins within the cell and allows waste to exit. When the plasma membrane is severely damaged, like in severe burns, the cell becomes highly permeable and substances flow freely in and out of the cell. For example, this is why severe burns weep, as proteins, ions, and fluids leave the damaged cells. Burn dressings are key for protecting these damaged cells from the environment. A thorough understanding of cellular structure and function will enable you to properly treat all of your patients, including those with cellular diseases and cell damage.
As the smallest living unit, they are very, very different in appearance. So on the left, right there, we have a skin cell. On the right, that is a neuron. You can see that their shape is very different, but each one of them has a barrier that keeps that cell from being invaded by things it doesn't want to be invaded by. There's rules on how to get into that cell. There's rules on how to get out of that cell. So in a nutshell, we're going to learn about the details, especially of that cell membrane and the organelles inside these guys that make these cells work and be alive. So there's a few generalized things that we're going to talk about with cells. Even though cells have different appearances, they all have these three things in common in general. There's always exceptions in science. So that cell membrane that I'm talking about that creates the rules, it's also called a plasma membrane. It's the craziest thing. On the inside of it and the outside of it, it carries an electrical charge. Isn't that crazy? It's flexible, so like if you poked on it, it would squish in and come back out. So, like the Pillsbury Doughboy's belly. It's not stiff like a plant cell wall. Then somewhere very, very protected with a double layer of the same thing that the plasma membrane is made of. So the plasma membrane is made of a phospholipid bilayer. Somewhere inside that cell, there is a very protected DNA center. It's got 2 layers of that phospholipid bilayer. Very, very protected. So wherever that DNA is, that's going to be called the nucleus. So there's going to be a nuclear membrane surrounding the nucleus with the DNA on the inside. And then the area between the plasma membrane and the nuclear membrane is going to be the cytoplasm. And this is where the majority of the organelles are floating around. They're floating in a little fluid called cytosol. This picture is showing, in general, the parts of a cell. So again, they're not all shaped oval or round. They vary. This right here is the control center. It's very well protecting the DNA. In this picture, the DNA is called chromatin, and we'll get into that detail later. But chromatin is the way that the DNA is laid out. So repetition is key to learning. Chromatin is the way our cells usually have the DNA arranged inside a nucleus. It's like a kitten got a hold of a bundle of yarn and knocked it all over, woo, into a skinny, unwound thread. You've probably heard the term chromosome before, and actually, we only bundle up the chromatin into chromosomes when it's time to go through mitosis. So 99% of the time, it's very thin and all willy-nilly all around on the inside of this nucleus. This is an organelle called a nucleolus. So this guy has his own job to do. Notice he is on the inside of the nucleus. He's going to help make ribosomal RNA, pieces of ribosomes, if you will, and we'll look at that in more detail in lab and lecture. Remember, lab and lecture go together. So now that we have the basics of the nucleus out of the way, where we're going to keep, this is your most protected part of your cell overall, because we don't want anybody to go and mess with our DNA. That's bad. So on the outside of the nuclear membrane, also called the nuclear envelope, boop-a-doop right there, on the outside of it and on the inside of the cell membrane, this is all the cytoplasm area. And the cytoplasm is made up of cytosol, the liquid that's on the inside of this cytoplasm in this area, and the organelles. So this is a mitochondria. That's one of the organelles. Smooth endoplasmic reticulum. That's another organelle. Rough endoplasmic reticulum. That's another organelle. Golgi apparatus, sometimes called a Golgi body. That's another organelle. So there's more. We'll do them in class. But whenever you're studying this for lab and lecture, this is a great picture to go back to. Whenever you hear the term extracellular materials, we're talking about things that are outside of a cell. So there is a tiny region between a cell and, let's say, a blood vessel, okay? There is a region outside of your blood cells, but still inside your vessel. That'd be plasma. Right? If you've worked in a hospital, you've drawn somebody's blood, or you've seen blood drawn or whatever, you can take that blood and spin it down, and all the cells go to the bottom, and the rest of it is fluid, where your oxygen's carried, where your, well, not your oxygen is going to be mostly on your red blood cells. Scratch that. Hormones. sodium chloride, potassium, so your electrolytes. It's going to have proteins, lots and lots of proteins, antibodies. You've heard of all those things. Those are all extracellular. So they're in your blood vessel, but separated from the cell. And then again, you've got fluids that are protecting, like your brain, a baby, amniotic fluid. So these are all fluids that are examples of fluids that are outside of a cell. Then we're going to have fluids that are actually made by a cell, cellular secretions. You're familiar with sweat. You're familiar with your child's snotty nose. You're familiar with smell some pizza and you start getting saliva in your mouth. You're familiar with all that. Some of these areas, like at the interstitial fluid, immediately outside of a cell, but not quite into a blood vessel yet, all of that area is held together by something called extracellular matrix. Most of it is something called connective tissue, and we'll get into details of that later. So the plasma membrane, also known as the cell membrane, is the guy that creates the rules that keeps things from going in and out of a cell without any sort of craziness. You must have rules. You must have boundaries to be alive. Chapter 1. So essentially what we're doing is we're separating that outside of the fluid cell, outside of the cell fluid, extracellular fluid, from the intracellular fluid, the stuff that's on the inside of the cell. And here is where I'm going to talk about what it's made of. Phospholipid bilayer. Bilayer means it's 2 layers of phospholipids. Then we're going to have some special proteins that are completely embedded in that phospholipid bilayer. They are going to be kind of like gatemen most of the time. Somebody that's guarding the gates, but opens the gates and lets things in and out if the cell says it's okay. Some of that phospholipid bilayer is going to have some molecules on it that serve as an identity marker. So it's called a glycocalyx. And you have a different glycocalyx from your parents, your spouse, your children. You have these little markers on your cells that say, this is me. And then cholesterol, I mentioned that in the other chapter with organics. Cholesterol actually is embedded in different areas of a cell membrane that allows for a little bit of flexibility, and it actually helps make it stronger. And then finally, some cell membranes, not all of them, epithelium especially, is going to have some cell junctions that kind of help hold cells together right next to each other. So here's a picture. This right here, little round things, are phosphate groups. And the things that are coming off of them are 2 fatty acids. Do you remember that from the organic section when we talked about lipids? You have triglycerides that are what you eat, but phospholipids are a huge importance in biology. So when I say it's a bilayer, here's a phosphate group with 2 heads and they're all lined up tail to tail. So the phosphate group is actually water-loving, hydrophilic. But remember, fatty acids, the lipid part, these are hydrophobic. What's passing by your cells? Fluid made of water. We're not oily people, we're watery people. And they're all happy. These phosphate heads love water. Same thing with this. This is the inside of the cell right here. Inside of your cell, got a lot of water, loves water. It's all good. There's no difficulty mixing. So the part that doesn't like water is enveloped on the inside. What this is going to do is anything else that loves water can't just go sneak on through here, usually. It can't because of these lipids right there, these fatty acid tails. These little things that we mentioned in also chapter 2, stop sign, stop sign, stop sign house. It's not really a stop sign because it's really, stop sign's a hexagon, whatever. That's cholesterol embedded in there. This guy right here is a protein. This guy's a protein with a little tag on it, a carb. Same thing. Here is a little bit of lipids attached to a carb right there. So these little things are partially what makes up your glycocalyx. Here's a little protein. He doesn't get to go all the way through. Whenever they go all the way through, that's an integral protein. Whenever it's just on the edge, that's called a peripheral protein.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So this is reiterating what I just said. The majority of that cell membrane, that plasma membrane, is made of phospholipids. The polar heads, the phosphate groups, actually have a charge. The fatty acid tails are nonpolar, and so they are hydrophobic, and they don't have a charge. So do you remember me saying at the first slide, I said the crazy thing about these cell membranes on the outside and the inside, they have a charge. And that charge is going to also be a huge player on what can come in and out of that cell. We're going to have some cholesterol, like I told you, increasing that membrane stability, makes it strong but flexible. And then we have these other tags that are sticking off of it that create that glycocalyx that's yourself. Some of it's fats, some of it's proteins, and they're all bound with a sugar sticking off of the surface. So this is a little picture again. This picture was in chapter 2 when we did the lipids, a phospholipid. So this guy right here is the phosphate group, and he has a charge, and he loves water. These little guys, oh look, he has a kink in it. What does that mean? Do you remember? So there's 2 fatty acids right there. Fat doesn't like water. So the way that they're lined up, tail to tail, that allows this part right here, because this is the outside of a cell, so that's extracellular fluid, perfectly happy with those phosphate groups. And in here, they didn't draw all the organelles or anything, but you know that there is cytosol in here, so that fluid, and that's okay because these guys like fluid. This part right here is the part that doesn't like water. But it's okay, they're hidden. So within that membrane, we have those proteins, and I already pointed out some go all the way through, and those are called integral proteins. Some are just on the edges. The picture just showed them being right on the inside, but sometimes they're just right on the outside. All of those guys are going to carry a function. So the integral protein, the huge thing about them is they are going to have usually a receptor for something, like a hormone or some sort of chemical, neurotransmitter, whatever. There's tons of functions that your book goes through, but I'm going to highlight especially the transport channels and carriers that let things in and out. Enzymes and the receptors we're going to see a little bit later in the chapter and in AMP2. So just keep in mind that these integral proteins have lots of different functions, and they help create the rules. They need to be told to open up sometimes to let things in. Depends on what kind of channel it is or carrier it is. And then the peripheral proteins, same thing. They actually have lots of different functions, and we're really not going to get into detail in them, but you should be able to recognize what a peripheral protein is. This is showing different pictures of how those integral proteins can work. So sometimes we're going to have things going from the outside of the cell to the inside of the cell. So that's going to be a channel. Sometimes they're very specific. I am just for water, or I am just for electrolytes. But we'll get into that later. Sometimes we're going to have an enzyme on the inside of the cell creating things because of this guy right here, creating things going on the inside of the cell. Sometimes we're going to have a receptor on the outside of the cell, and when some sort of key shows up and fits in here, that is going to cause things to happen on the inside of the cell. Sometimes those guys help cells stick together, and sometimes they're actually going to serve as an attachment for the skeleton that's on the inside of the cell. We have things that help on the inside that help keep that cell maybe from getting squished, or maybe there's things that are walking around on that skeleton. It's the craziest thing. There is a video in maybe Lanyap called The Cytoskeleton Highway. So awesome. Go ahead and watch it. So the glycocalyx is going to be some sugars and then probably also holding on to something else, but it says that you are you, okay? That's called the glycocalyx. The immune system uses that glycocalyx whenever you're invaded by a virus. So let's say the flu, so flu is a virus, not alive. He must get inside of your cells to continue to reproduce. He uses your stuff to reproduce. So viruses are not classified as live creatures. Well, when he gets into one of your cells, your cell plucks all the glycocalyx off of it. That cell membrane is continuously changing. It is amazing. So when he plucks that glycocalyx off, because he's been invaded, when your immune cells are passing by, they're always on patrol. Hey, who are you? I don't see your glycocalyx. You do not belong to this person. I'm going to kill you. Now, does it matter? Because he's going to die anyway, right? He's got the flu virus in him. He's going to die. Yes, because what that does is it hopefully buys you some time to get better before the flu virus takes over your whole body. Sometimes we'll have cells that sneak by. So cancer cells are an example of, they are your cells that have gone rogue, and they are constantly changing so fast they confuse those patrollers. They're like, well, they're still going through mitosis, so I'll come back and check on you later. And that's how one way cancer can keep moving on. Okay, so I said some of these cells actually have a way to grab a hold of another cell. these are 3 different ways that those cells can bind to each other. So one of them is called a tight junction, one of them is called a desmosome, and the other one's called a gap junction. A tight junction is just like it sounds. It's usually towards the apical surface of a cell, so not where the cell is setting on the beneath tissue, but the part that's usually going to be free. So it's not attached to anything at the apical surface at the top. You've heard of an apex predator? They're at the top of the food chain, great white shark. Okay, the apical surface is not attached to anything. And so at the area of closer to the apical surface, right here, these guys bind to each other, bind to each other, bind to each other, bind to each other. You may know this already, that your skin changes over once a month. So you can take your fingernails and scratch your arm, and you're going to get some skin cells that we're fixing to slough off today anyway. They don't come off one at a time. They come off in sheets because of these tight junctions. Very helpful for waterproofing your body, by the way. For, if you get rained on, does the rain absorb into your skin? No, rolls off. Thank you, tight junctions. Desmosomes, what they're going to do is they're like zippers. They're about midway between the cells. And what their purpose is to prevent tearing under tension. So if you push on your skin, there's some little zippers that are keeping them together. Your heart muscle cells, very important. They're all zipped together. That would be bad if your heart muscle cells separated, would it not? And then finally, cell junctions called gap junctions are going to be closer to the base of the cell. So not the free end, but the part that's setting on something else. These little gap junctions are like tunnels, and the purpose of them is to allow cells to exchange stuff. So, hey, I've got plenty of water. You need some water? Here you go. Hey, I've got some salt. You need some salt? I got excess. You want some salt? It's going to pass back and through. Sugar, I got some sugar. We're going to diffuse through, and we're going to make it all evenly spread out. And then even sometimes electricity can pass from them, from one to another. So, to get through the membrane, what are we going to do about that? That plasma membrane is called selectively permeable because some things can get through. It depends on what it is. Some things need one of those integral proteins, a transport channel or carrier pathway. So, if I am hydrophobic, if I am nonpolar, I don't have that hard of a time getting through those tails. Example, alcohol. Alcohol, if it ends in an OL, it probably has a cholesterol base or steroidal base. And so if it ends in an OL, it's more likely to be hydrophobic. No problem sneaking through those tails, gets into your cell, and of course, if you have enough alcohol, makes you do goofy things. Large molecules are not going to get through. Charged particles are not going to get through. Water, even though, of course, water is water-loving, a single water molecule is tiny. So sometimes he can dodge those phospholipid tails, just like, who was it? Dory and Marvin in the jellyfish thing in Nemo. Water can kind of do that because he's so small and actually get through. But a lot of times he is going to use a protein channel. It's actually going to be called an aquaporin and just go through that channel that way. And then he doesn't have to even try and dodge those phospholipid tails / jellyfish tails. All right. So how do we get through the plasma membrane? Well, sometimes you can just diffuse through, and we're going to go through diffusion in a minute. It's a large portion of lab. The next thing that we're going to do is use active transport, which means that it requires ATP for it to happen. Let's do the no energy required part, that passive membrane transport. So if you see passive, that means no energy required. Everything that we do in lab is going to be passive. So they all involve a type of diffusion. What does that mean? That means that we have molecules that are moving from an area of high concentration to low concentration. Another name of that is called moving down the concentration gradient. So if you can imagine, you're sitting at a bench and one end of the bench is getting really crowded and nobody is at the far end of the bench. Scoot on down. Just move down the concentration gradient from high concentration of people to low concentration of people. You're just going to scoot on down to make everything evenly spaced out. So that's essentially, in a nutshell, what diffusion is. We have molecules that are in random constant motion, so like a kinetic kind of energy, not ATP energy. One thing in lab, that Brownian motion that we've been through, is that some biologist named Brownian was looking at pollen under a microscope, and he noticed that the pollen was moving. So he had it on a wet mount, which means he takes pollen, puts it in a little dollop of water, puts a slide on it, or a cover slip on it, and looks at it. And all of the pollens were jiggling. What the heck? Why is my pollen moving? The pollen is not alive. What he figured out, though, was that water molecules, which cannot be seen, were bumping into his pollen. And the pollen's bumping into his water molecules. So what's happening is matter is always in this kind of random wiggle room, even like your desk. You know, there's potential energy in there, stored energy. Those molecules have the tiniest amount of motion, even though they're compacted all together. So what's happening is when you, what he was seeing, how do I say that? What he was seeing was this movement of molecules. Even though he couldn't see the water was bumping into his pollens, and so therefore the pollens were wiggling just a little bit, but enough that you're like, how about that? You too can do that yourself at home if you have a microscope. I'm sure you can YouTube it. Okay, so when let's do diffusion with some room spray. You walk in and your brother just got home from football practice, took off his shoes and left it right next to your desk. Whew, that's bad. So you spray room spray to help your desk area smell better. What did you spray? You sprayed a bunch of molecules that smelled good. Well, probably in about 5 minutes or so, you're not going to smell that just at your desk area. You are going to smell it maybe 10, 15 feet away. Because when it came out of that can, that room spray came out of the can, it was in high concentration. But he wants to move. He wants to be evenly distributed. That's the law of nature. So he's going to move from an area of high concentration, your desk, with the stinky shoes next to it. And it's going to move across the room so that those smell good molecules spread out. You know, chances are you could probably smell your brother's shoes from 10 feet away too. But just got more concentrated as you got closer to his shoes. Those particles are trying to move about. So what I just described there is simple diffusion. There's no barriers. It's just going from high concentration to low concentration. This is what we're going to do in lab. We're going to put some dye inside of some water, and we're going to watch what was clear water become the color of the dye. Just random Brownian movement, going to move from an area of high concentration to low concentration. And finally, the entire water will be the color of the dye. So, small nonpolar substances that I said nonpolar doesn't have any problem getting through those phospholipid tails. They're very easily going to diffuse from an area of high concentration to low concentration into or out of the cell. Oxygen, carbon dioxide, very tiny. And isn't that good? Don't you want the oxygen to easily get into your cell? Don't you want the carbon dioxide to easily get out of the cell? Other things would be steroidal hormones. They can get in because steroids are lipid category, remember from chapter 2. And so they can get in easily. Alcohol, get in easily. And then very, very, very small polar substances, especially water, can get in small amounts. They'd rather use a channel, but they can sneak through.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So this is showing how on the outside of a cell we've got some lipid droplets, and they have no problem simply squeezing past those phospholipid bilayer through the fatty acid tails. Large guys, not so much. If they are large, if they are, of course, polar, so hydrophilic, or if they are charged, they're going to have to use one of those integral proteins. Some examples would be glucose, amino acids, so the smallest block of a protein, and then ions, remember, are charged particles. So whenever something needs help, that's called facilitated diffusion. Facilitate means to help. I'm facilitating you to learn anatomy and physiology, but in the end, it's all up to you, right? Okay. So a channel mediated means that these guys aren't terribly picky. They're going to be opening up or allowing water to get through and some ions to get through. Sometimes they stay open, and those are called leakage channels. Doesn't that make sense? Sometimes they're gated, so they open and close based on maybe some sort of chemical that tells the gate to open or electricity that tells the gate to open. Okay, more specific from channel mediated is a carrier mediated. So what this means is that we're going to be, for very specific molecules, something on the outside of the cell actually physically lock and key binds to that integral protein. And in biology, when stuff binds to stuff, stuff changes shape. So when it changes shape, it actually allows something to come in. The thing about these guys, sometimes they get saturated. And so if you get too much, you're going to have spilling out, such as sugar. You know, if you have diabetes uncontrolled, you got too much sugar floating around, it's going to get to the point where you can't get any more sugar into, well, I should say sugar in general. If you had 12 Snickers bars, they're not going to all rush into the cell because you don't need that much sugar. So your body's going to store it some other way. This is showing that lock and key mechanism. So there's that carrier-mediated protein. And you can see he's staying locked on the cell side, the inside of the cell side. But if the right thing comes and binds to it, it's going to change shape and allow that thing in. Very important here, if you get a bad burn and you damage your skin, you've just broken the rules for your skin cells. That's true for anything, a poison or whatever. Something damages that plasma membrane, then things are going to go in and out of that cell willy-nilly, and actually that's how a lot of poisons work, toxins work. Osmosis is still diffusion, but we're talking specifically about water here. Water loves to follow things. It says, "Oh my gosh, I'm so concentrated in myself, and it looks like you need a little bit of me because you don't have as much water as I do." And so water follows things from an area of high water concentration to low water concentration. It's going to move across a semi-permeable membrane to make everything an equal amount of water. So it's the same idea as molecules moving from high concentration to low concentration, but this is specifically water moving from a high water concentration to a low water concentration. Also on this, we're talking about specifically through a selectively permeable membrane. So when I did the perfume, or whatever, room spray, that's not going through a membrane, it's just floating through air. That's simple diffusion. Osmosis is still a type of diffusion, but it is water moving from high concentration to low concentration, and it's going through a selectively permeable membrane. So on this example, if I have 5% sodium chloride on one side, 10%, I should say salt water. How about that? 5% sodium chloride is 5% salt in 95% of water. 10% sodium chloride is 10% salt in 90% of water. You see? So which one has less water? The 10% sodium chloride. So the water, if you put a membrane between a semi-permeable membrane between this 5% solution and this 10% solution, water is going to leave the 5% solution and go towards the 10% solution because the 10% solution has less water. It's got more solutes, less solvent. Water, universal solvent. Remember that? So moving from high water concentration to low water concentration, it's going to move to the 10% sodium chloride side. When we're talking about a 5% solution, 10% solution, that's the osmolarity of a solution. So if, like that 10% solution has more solutes, that's called a hypertonic solution to the 5%. It had more solutes, so that 10% sodium chloride was hypertonic to the 5% solution. This is one of those things that when you really focus on it, it makes complete sense. But you need to study this. It's not hard. It just requires repetition, repetition, repetition. In lab, the way that we demonstrate it is we have 2 Karo syrups sitting in some distilled water. The Karo syrup is bagged up in a semi-permeable membrane. One of them is all Karo syrup. The other one's been diluted, 50% Karo syrup and 50% water. So clearly the straight Karo syrup has less water, right? We added water to the other one. So when we put these guys in a beaker of distilled water, the water is going to move into those bags because neither of those bags are 100% water. They both have some sort of solute in them, Karo syrup. But the one that is 100% Karo syrup is going to be more attractive because it's like, wow, you don't have any water. I need to get in there, and I need to get a lot in there. The other one has 50% Karo syrup and 50% water. So it's still going to move in there because the bag's not full of water. So water moving from high water concentration to low concentration is still going to go into that bag, but not as much as the one that's 100% Karo syrup. The way that you can see that the water is going in is those bags are also attached to a thistle tube. So the water rises up the tube. Please refer to lab. Once everything is equaled out, so the same amount of water is on one side as the other side, then movement of water will quit. So this is showing a tube right here with a semi-permeable membrane. Maybe we could say this is the 10% salt water. So that's all the red or salt dots. And then this is 5% water. So more water, less salt. It's only 5% salt. So the water is going to move from high concentration to low concentration. Look what's going to happen. I have enough water in here that I have made this a 7.5% solution, and this is a 7.5% solution. They evened out. The water evened out. That's what's going to happen in a cell. We don't want a cell to get too full of water, or it'll pop. We don't want a cell to lose too much water. It'll shrivel up and die. So when you put something in the body, like a solution of IV fluids, you're going to want to make sure that whatever you put in is the same tonicity as everything in your body. That would be isotonic, because otherwise things are going to fill up a cell and make it pop. If it is going to be a hypotonic solution that you put in there, water's going to move from high concentration to low concentration and fill up all your cells, and they're going to break. That's called lysing. If you put in a super hypertonic solution, then water is going to leave the cell and go into your vessels. So your cells are going to shrink. That's called crenation. This is a picture of what happens. There's a YouTube link for you to watch.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypotonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid, and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell, and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell, and it will burst open, or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in the shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, water rushes out of the cell and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored.
And then in lab, finally, we're going to go through these different things that change the speed of diffusion. The more concentrated something is, the faster it's going to diffuse. The smaller a molecule is, the quicker it can move, so the faster it'll diffuse. And then finally, temperature. The hotter something is, the faster it will diffuse. And we're going to do all those things and show you that in lab.